Assalamu alaikum. Now I would like to talk about glycolysis. Before jumping into glycolysis, I would like to discuss about the sites of the metabolism. First of all, uh, mitochondria. In mitochondria, there are some pathways which occurs in the mitochondria, and you can remember it by A, B, C, OK. That means acetyl-CoA production, that means beta oxidation, that means citric acid cycle or TCA citric acid cycle or TCA cycle, O for oxidative phosphorylation, and K for ketogenesis. Okay, so should I repeat it again? What are the sites of metabolism of these pathways? The acetyl-CoA production, beta oxidation, citric acid cycle, oxidative phosphorylation, and ketogenesis. These occurs in mitochondria. Then cytoplasm, and both you can remember by HUG, that means heme synthesis, urea cycle, and gluconeogenesis. Heme synthesis, urea cycle, and gluconeogenesis. So if you remember these two things, that mitochondria and both, you can easily answer the rest of the should be cytoplasm. And in cytoplasm there is glycolysis, there is HMP shunt, there is synthesis of cholesterol, there is protein synthesis, fatty acid synthesis, and nucleotide. Okay, now I would like to discuss about the pathway of glycolysis. In glycolysis, first of all, glucose is converted into glucose 6-phosphate, and glucose 6-phosphate is converted into fructose 6-phosphate, and in fructose 6-phosphate is converted into fructose 1,6-bisphosphate, fructose 1,6-bisphosphate. From glucose to glucose 6-phosphate, there is an enzyme called gluco or hexokinase. Okay, glucokinase found only in liver. Glucokinase found only in liver. On the other hand, hexokinase found in most of the tissues except liver and beta cells of the pancreas, except liver and beta cells. So glucokinase is found in liver and beta cell, and hexokinase is found in rest of the cell except liver and pancreatic beta cell. In glucokinase there is a higher Km, higher Vmax, and Km is higher in glucokinase. On the other hand, lower in hexokinase. In glucokinase is induced by insulin. It is induced by insulin. Glucokinase is induced by insulin. On the other hand, hexokinase is not induced by insulin. And the last is feedback inhibited by glucose 6-phosphatase. Glucokinase is no and hexokinase is yes. And these are the difference between glucokinase and hexokinase. So where I was, glucose is converted into glucose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate. Then fructose 6-phosphate is further converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone and then these glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate or 3-phosphoglyceric acid, 3-phosphoglycerate. Then the 3-phosphoglycerate is further converted into 2-phosphoglycerate. 2-phosphoglycerate is further converted into phosphoenol pyruvate. With the help of pyruvate kinase. And pyruvate is further converted into lactate. With the help of lactate dehydrogenase. Pyruvate is converted into lactate with the help of lactate dehydrogenase. In here there are something you have to remember, that pyruvate kinase deficiency. This is a thing you have to remember, that in pyruvate kinase deficiency and glucose 6-phosphate dehydrogenase deficiency. In both deficiency, in glucose 6-phosphate dehydrogenase deficiency and pyruvate kinase deficiency, there is chronic hemolysis. In both cases there is chronic hemolysis. So how can you differentiate these two things? In pyruvate kinase and there is increased 2,3 BPG and there is absence of Hinge bodies. In pyruvate kinase there is absence of Hinge bodies. On the other hand, in glucose 6-phosphate dehydrogenase, Hinge bodies are characteristic feature in glucose 6-phosphate dehydrogenase. So you can remember that in these two cases, in pyruvate kinase deficiency and glucose 6-phosphate dehydrogenase, in both cases there is chronic hemolysis, there is increased 2,3 BPG, but in pyruvate kinase there is absence of Hinge bodies. On the other hand, in glucose 6-phosphate dehydrogenase deficiency, there is presence of Hinge bodies, which is most characteristic features in glucose 6-phosphate dehydrogenase deficiency. Okay, there are some other things we have to remember, that these from glucose there is pyruvate, and this pyruvate can convert into fourth thing. This pyruvate can be converted into fourth thing. This pyruvate can go into can form lactate. This pyruvate can convert into alanine. This pyruvate can be converted into acetyl-CoA, and finally this pyruvate can be converted into oxaloacetate. So pyruvate can be converted into lactate, pyruvate can be converted into alanine, pyruvate can be converted into acetyl-CoA, and pyruvate can be converted into oxaloacetate. Pyruvate can be converted into lactate with the help of pyruvate with the help of lactate dehydrogenase enzyme, and as a vitamin you need B3 in such case, and pyruvate can be further converted into alanine with the help of ALT. Okay, sorry for this hassle. Now, pyruvate, pyruvate can be converted into acetyl CoA with the help of pyruvate dehydrogenase. Pyruvate can be converted into acetyl CoA with the help of pyruvate dehydrogenase. And in here you need a 5 coenzyme. We will talk it later when we will discuss about pyruvate dehydrogenase complex. So pyruvate can be converted to lactate with the help of lactate dehydrogenase. In here vitamin B3 helps. In here vitamin B3 helps, and pyruvate can be converted into alanine. In here vitamin B6 helps. And pyruvate can be converted into oxaloacetate with the help of pyruvate carboxykinase. And in here pyruvate carboxylase. In here pyruvate carboxylase. In here vitamin B7 helps. And last but not the least, that pyruvate is converted into acetyl CoA with the help of pyruvate dehydrogenase. Okay, pyruvate dehydrogenase complex. That pyruvate is converted into acetyl CoA. Pyruvate is converted into acetyl CoA, and in here we need 5 enzymes. There is a mnemonic saying tender, loving, care for Nancy. Tender means thiamine. Loving, that means lipoic acid. Tender, loving, care, that means as CoA. For means FAD, and Nancy means NAD. This 5 enzyme is needed in this pyruvate dehydrogenase complex. Pyruvate is converted into acetyl CoA with the help of pyruvate dehydrogenase, and in here you need these 5 enzymes. These 5 coenzymes: tender, loving, care for, Nancy, thiamine, lipoic acid, CoA, FAD, and NAD. And another thing you have to remember, that thiamine is vitamin B1 and CoA is vitamin B5, and FAD is vitamin B2, that means riboflavin, and NAD is vitamin B3. Niacin. So you need 1, 2, 3, 5, these 4 vitamins as a core factor in pyruvate dehydrogenase complex. Now I would like to talk about galactose metabolism. Okay, in galactose metabolism, what you have to remember, that lactose is converted into glucose and galactose. Lactose is converted into glucose and galactose with the help of lactase enzyme. If there is deficiency of lactase enzyme, it will cause lactate deficiency. Lactase deficiency, and in lactase deficiency the features is BCD, that means bloating, that means cramps, and D for diarrhea. Bloating, cramps, and diarrhea. Then galactose is converted into further converted into galactose 1-phosphate, and galactose 1-phosphate is further converted into glucose 1-phosphate. Galactose is converted into galactose 1-phosphate with the help of galactose is converted into galactose 1-phosphate. If there is a deficiency of these enzyme, that galactokinase, If there is deficiency of galactokinase, it will just cause cataract. And how it cause cataract? When there is deficiency of this galactokinase, then these galactose will accumulate. Galactose will accumulate. This accumulated galactose goes to the lens and converted into galactitol with the help of aldose reductase. So you can remember it easily that this galactose, if there is deficiency of galactokinase, this galactose is converted into galactitol with the help of aldose reductase, and galactitol trapped in the lens and cause swelling and cataract. And this is the pathway of having cataract in galactose deficiency, in galactokinase deficiency. And we know that galactose is further converted into galactose 1-phosphate is converted into glucose 1-phosphate with the help of what? With the help of galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase. Uridyl transferase. Galactose 1-phosphate uridyl transferase. In here, along with cataract, you will found there is mental retardation. There is vomiting, diarrhea. There is hyperbilirubinemia. There is vomiting, diarrhea, hyperbilirubinemia. There is vomiting, diarrhea, and hyperbilirubinemia. Hyperbilirubinemia, and this is called classical galactosemia. In classical galactosemia, the patient, this type of patient, have the tendency of having E. coli infection. The E. coli infection can predispose to E. coli sepsis in neonates. It can cause hepatomegaly, it can cause vomiting, diarrhea, it can cause mental retardation, and this is the high yield point. In galactokinase deficiency, there is no mental retardation, but there is cataract. But in classical galactosemia, along with cataract, there is hepatomegaly, there is hyperbilirubinemia, there is mental retardation, and you have to remember this in a very good manner. Next, now fructose metabolism problem. We know that sucrose is converted into glucose and fructose. Sucrose is converted into glucose and fructose with the help of what enzyme? Sucrase enzyme. Then this fructose is converted into fructose 1-phosphate. Fructose is converted into fructose 1-phosphate with the help of fructokinase. If there is deficiency of this enzyme, it causes essential fructose urine, which is benign, and fructose 1-phosphate is further converted into dihydroxyacetone phosphate and glyceraldehyde, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate with the help of aldolase, B. In aldolase B deficiency, there is lethargy, vomiting, diarrhea, hypoglycemia, hyperbilirubinemia, hypoglycemia, there is a renal proximal tubule defect, there is Fanconi anemia, there is renal proximal tubule defect, These things you have to remember, and it happens if the baby is fed in the early age, or in the early age the baby is fed fruit juice, honey, and this kind of thing in early ages. In these cases there is problem in fructose metabolism, which is called hereditary fructose intolerance. Hereditary fructose intolerance due to deficiency of aldolase B enzyme. So for this kind of patient, they should intake less fructose and sucrose, that means glucose and fructose, and this is the way they should live. Now, TCA cycle. In TCA cycle, what you have to remember, that TCA cycle, also known as a Krebs cycle. What happens in TCA cycle? First of all, pyruvate is converted into acetyl CoA. Pyruvate is converted into acetyl. Pyruvate is 3 carbon, acetyl CoA is 2 carbon. This acetyl CoA mixed with oxaloacetate, which has 4 carbon. Oxaloacetate, which has 4 carbon, and forms citrate and forms the 6 carbon citrate. Then citrate is converted into alpha ketoglutarate. Citrate is converted into alpha ketoglutarate. Alpha ketoglutarate is converted into succinyl CoA. Succinyl CoA is converted into succinate, fumarate, and malate. Succinate, fumarate, and malate, and then this malate again converted into oxaloacetate, and this way the pathway goes, and this pathway goes. In alpha ketoglutarate dehydrogenase complex, it requires the same 5 coenzyme, which is called tender loving care for Nancy. So pyruvate dehydrogenase complex, we need tender loving care for Nancy, and alpha ketoglutarate dehydrogenase. The pathway in between alpha ketoglutarate to succinyl CoA, is alpha ketoglutarate to succinyl CoA. You need alpha ketoglutarate dehydrogenase. You need, in this case, you need that 6 enzyme to tender loving care for Nancy, and the other pyruvate carboxylase, that means pyruvate carboxylase from pyruvate to oxaloacetate. In these cases you need biotin. Okay, then ethanol metabolism. In ethanol metabolism, what happens, that ethanol is converted into acetaldehyde, and then this acetaldehyde is converted into acetate. Ethanol converted into acetaldehyde with the help of alcohol dehydrogenase. Acetaldehyde is converted into acetate with the help of acetaldehyde dehydrogenase. Alcohol dehydrogenase can be blocked by a drug called fomepizole, and acetaldehyde is converted into acetaldehyde dehydrogenase, and this can be blocked by disulfiram, and this disulfiram discourages drinking. This disulfiram discourages drinking. And that's all about glycolysis metabolism. Inshallah, you will be benefited.